Alright guys, welcome back to Underground Science, and in this video we'll be looking at the loop of Henle. Alright, so let's go and get started. So in the proximal convoluted tubule, we said that we reabsorb different substances, right, different nutrients. Now the loop of Henle will act as, well, the one part of the loop, right, so let's just jump right into it. So the descending loop of Henle, right, which is this part, the descending loop of Henle, right, will actually be permeable to water. So the descending loop of Henle is responsible for taking water out of the lumen and into the interstitial space, all right, of the kidneys, of the nephron, so the renal medulla area, all right, and this part is going to be known as a countercurrent exchange system. We'll see that when we talk about how over here, right, in the descending loop of Henle, we release a lot of water into the interstitial space of the renal medulla, and then in the ascending loop of Henle, what actually happens is that these the cells right here are impermeable to water, so there no water can leave or enter. All right, and only solutes will then be leaving the cells of the ascending loop of Henle cells. All right. So let's go ahead and get started. So over here, we have to know that in the descending loop of Henle, we have a bunch of water channels called aquaporins. All right, so we'll have some aquaporins here, right? So we'll have aquaporin there, right? We'll have aquaporins here. So let's still go draw it quick like that. Aquaporins here, and these in particular, these are aquaporin type 1 channels. All right, and here there's going to be a lot of water flowing down here. So all this water is actually going to be permeable in the descending loop of Henle. Okay, so all this water will actually leave the descending loop of Henle, sorry about that, couldn't speak there, and will actually enter the interstitial space of our renal medulla. So we're going to have a bunch of water here. And what's going to happen is that as water leaves, right, solutes aren't leaving. So that means as we go down the descending loop of Henle, the lumen, right, over here is starting to get very salty. So it's super salty, around 1200 milliosmoles. But that's for later, all right? Let's just look at the basic idea right now. So as water leaves, we have more and more solute buildup, and we get very salty here. All right, and because of all the sodium, potassium, chloride anions that are building up, right? However, once we start going up the ascending loop of Henle, right, in our ascending loop of Henle cells, what happens is that if we zoom in to one of our cells over here, right, if we go ahead and zoom into one of our cells here, so let's say this is an ascending loop of Henle cell, all right, and what happens, so let's go and write this as the interstitial space is over here. So this is the interstitial space, all right, and then we'll label this part, right, as the lumen. So this is the lumen of our ascending loop of Henle. So this is the lumen of our ascending loop of Henle. And so what happens is that we have these transporters here on the apical side, right, of our ascending loop of Henle cell near the lumen, and these transporters allow all these solutes to get transported into our cell and then out into the interstitial space through sodium-potassium pumps, ATPase pumps, all right? So we're going to have all of these sodium. So what we're going to take is that we're going to take one sodium, and we're actually going to take 2 chloride anions and 1 potassium anion, or cation, right? So again, we're going to take 2 chloride anions, 1 potassium cation, and 1 sodium cation, and we're going to transport all 3 of these solutes, right, into the cell, into the ascending loop of Henle cell. And from here, these solutes can actually enter the interstitial space, all right, particularly through a sodium-potassium ATPase transporter. So if we were to be specific about it, right, we also have a sodium-potassium ATPase transporter where we transport sodium from low concentration, which is inside the cell, to high concentration, which is outside the cell, right? So this is actually 3. and we transport potassium from low concentration. Okay, so let's choose a different color, from low concentration, which is in the interstitial space, to high concentration, which is inside the cell. And because we're doing low to high concentration, we need ATP for this, all right, which will get broken down into ADP and an inorganic phosphate. All right, and in particular, this is going to be our ascending loop of Henle cell, or our thick ascending loop of Henle cell. All right, so our thick ascending loop of Henle cell. All right, so our thick ascending loop of Henle is this part, okay, where it goes thicker. And this is our thin ascending loop of Henle. All right, so then what this is gonna result in is a bunch of sodium, right, bunch of chloride anions, but a bunch of potassium anions, right, building up here. So that's why they say the renal medulla is super salty. The loop of Henle is in the renal medulla, okay, and it's very salty because of all these solutes. And this countercurrent exchange mechanism is so cool that because all these solutes are constantly entering the interstitial space, it drives more and more water also. So this is passive transport, but this is active transport. And it works together by pulling more water in, right? And we'll also see a branch of the efferent arteriole, right? Especially in our juxtamedullary nephrons. So our juxtamedullary nephrons and cortical nephrons, right? Those are the two main types we'll talk about in a future video. But for this video, just know that a juxtamedullary nephron actually has a large loop of Henle, all right? And the efferent arteriole branches into the vasa recta. So if we looked at the vasa recta, right? If we looked at the vasa recta, we would see that we have the venule side of, it's kind of like a capillary bed. So we would see the venule side, right? So let me color this in more. And then we would see the capillary side on this side, right? And what's going to happen is that the vasa recta will supply nutrients to the renal medulla, or the loop of Henle, and also it's going to try to pick up some of these solutes in water, right? And it can actually, the venule side can actually branch together and form, and go branch together and form into, or dump into, I should say, into the renal vein. Okay, but this is in juxtamedullary nephrons. All right, so in general, juxtamedullary nephrons have a large loop of Henle, and the efferent arteriole branches into the vasa recta. So right here, this whole capillary bed structure, right, is going to be the vasa recta. Okay, and in cortical nephrons that have a short loop of Henle and actually a large proximal convoluted tubule and distal convoluted tubules, right, the efferent arteriole will branch into peritubular capillaries. All right, but sometimes you can see peritubular capillaries here also that can help in picking up some of this water and solutes and putting it back into the blood. All right, and the peritubular capillaries, especially in cortical nephrons, will then join in together and dump into the renal vein. Okay, but yeah, so I'm sorry, it was probably a little confusing, but because we haven't really talked about juxtamedullary and cortical nephrons, but we will in a future video. Okay, but just know for now that if we say there's a vasa recta here, a vasa recta's structure is we have a venule side, a capillary side, and supplying nutrients to our loop of Henle in our renal medulla. All right, and we can supply nutrients here, and it can play a role in countercurrent exchange by absorbing some of this water and sodium, maybe even releasing some of it and actually taking some of this water and sodium and chloride anions and potassium, joining it in all with the renal vein, right? So all these venule side of our vasa recta can join together into the renal vein. Okay, and then that can go back to the heart and dumped into circulation eventually. All right, so yeah, that's the basic idea, the loop of Henle in our renal medulla. So let's go and write that down. So the loop of Henle is actually in our renal medulla. All right, so yeah, I hope this video made sense to you guys. Thank you guys for watching, and make sure to tune into my future videos where I'll talk about more cool topics about biochem, physics, and chem. Don't forget to hit that notification bell, like, and subscribe. See you guys later, and stay safe.